Hello, everyone. Today we're going to go over what's actually not too much new content, but there's a lot of really subtle differences between some different kinds of graphs and ways of presenting the data for the chi-square test for independence, and also the logic of why we make the calculation for the expected values exactly how we do. So it's important to pay close attention, and you may need to listen to the video a couple times, but this is the only video for the whole topic. And then I have an example problem worked out step by step at the very end. So that one will be really important for you to watch all the way through and to work along. So make sure you're understanding all the steps. Okay, let's get started. All right, so our topic today is the chi-square test for independence. And we're going to assume that we've taken a sample, a single sample, from a population. The sample was randomly collected, and the individuals are independent from one another. We're going to evaluate each individual on two categorical variables. So we're going to look at them and we're going to say they are, brown versus blue, or they are one particular species or a different species. And then we're going to look at another variable. We think that these variables might be related to one another. In the same way, for a quantitative variable, we're interested in knowing when we do linear regression, if those two variables have some relationship with one another. The null hypothesis for our test is going to be that the variables are indeed independent, all right? So if we reject the null, then there is some relationship between the variables. And so knowing one thing about each individual will tell us something about their status on that other variable, all right? For a robust ability of the test to get the answer right, ideally we have a total sample size of over 50, and each cell count is at least 5. A little more sophisticated version of this you'll find in advanced textbooks that at least 20% of the cells in the table need to have greater than 5. And in a lot of our examples, we aren't going to have sample size, but they're just practice problems, and so we're not so worried. Finally, the chi-square test for independence is a non-parametric test, whereas in some situations, the chi-square test of goodness of fit is a parametric test because we're checking to see if our data match to some distribution, right? So, but the test for independence doesn't assume that the data on the individuals matches a particular distribution. We're just asking if they're independent from one another. Now, the fact that we use the chi-square distribution as the test statistic, and we find our p-value using it, isn't have anything to do with a distribution that we're expecting our variable to match to. All right, it is our test statistic that we calculate that is matching to that distribution, and it in fact has to, so that we can find the probability by finding the area under that function. All right. So when we do have a situation where we're going to apply the chi-squared test for independence, we're going to have these two quantitative variables, and we want to know if there's a relationship between them. So you'll typically see the data presented in a two-way table, and we made bar charts early in the semester using this format. And it's pretty easy to make these graphs. You just highlight the data table, including the labels, and it'll make you a nice bar chart. It'll put the variable that is on the rows into the legend, and then it will put the other variable on the x-axis. Okay, so notice that these data are showing the number of people who were wearing a helmet in a bicycle accident and that had head injury, or that had another injury that wasn't a head injury, versus no helmet. Did they have a head injury versus another injury? All right, so we need to also have, so these are each of the categories that we see in this little table. But the variable is actually something that summarizes these two possible cases, right? So helmet versus no helmet, those could be bicyclist safety choice, helmet or no helmet, and head injury versus other injury could be bicycle accident injuries, right? So there's an overall category or label that should be at the top and side of this table as well. I've put them on the graph. Bicycle accident injury. Well, we have two kinds, head injury and other injury. All right, so if you're thinking to yourself, oh, wait a minute, it looks like there's fewer head injuries among the no helmet folks. Than there are among the bicycle folks, right? So notice that what we have on the y-axis here is count. So that's how many individuals we counted up in our sample, right? So the ambulance drivers had some little form they filled out, or as they reported their statistics that they could be tallied by researchers. Did they was the patient a bicycle accident injury? Yes, no. Were they wearing a helmet? Yes, no. Did they have a head injury or another injury? Um, so look at these totals though, right? So out of all of the accidents, over 5,000 were wearing a helmet, but only 1,600 were wearing no helmet. And we can see this on the bar over here, these other injuries. There's way more other injuries than for helmet versus no helmet. And that is reflected in the fact that there's just a much larger sample size of the folks wearing a helmet, which is good. They're smart people in Australia where these data were collected. So what can happen when you have a big difference in sample size between your variables here is that you can see results like this, where they seem like not to make too much sense. Okay, so what we're curious about when we're looking at this independence or dependence of helmet, wearing a helmet or not versus head injury, is we want to compare the head injuries uh, of individuals wearing a helmet versus no helmet. But if we have a big difference in sample size, look at the other injury compared to the head injury. All right, we've got a lot more other injuries because we have a lot more cyclists, right? And the proportion of other injury to head injury, right, is much bigger. So there's a bigger difference between how many other injuries and how many head injuries there are. When we look at the other injuries versus head injuries for no helmet, right, because the sample size is smaller, these bars are lower, right? But it looks like the proportions are different, right? So kind of look at the slope of the line connecting these two bars versus the one with head injury and other injury. All right, so if our sample sizes are equal or close to equal between our two different conditions that we want to compare, it'll be fine to make a graph just showing count, all right? But if there's a big difference between these two sample sizes, then we want to actually scale the bars on the graph. So that is reflected, right? And how we're going to do that, we're going to take the number of head injuries, the number of other injuries, and we're going to divide by the total, right? So it's convention to put the variable that you're really interested in comparing. And we really want to know what the difference is between helmet and no helmet, right? We're not really so concerned about the difference between head injury and other injury, right? So we really want to highlight this comparison. And with the helmet versus no helmet in our rows, all right? So rows are going to go across from left to right. And the columns, all right, columns go up and down like the Greek columns that hold buildings up. Those are going to be our sort of our response variable, right? So in Excel, it's always going to put this row variable, it's going to put it in the legend, and it's going to put the other one on the x-axis. All right, so notice how this makes it easy to compare helmet versus no helmet. All right, helmet versus no helmet. All right, and because we want to compare helmet versus no helmet, we want to divide the head injuries for helmet divided by the total helmet, right? So we want to rescale these values, and we want to rescale the no helmet values, right? So I've only put the row totals on here to help you see what I'm doing. Right, and so we're going to divide 327 by 5087, and that gives us 0.07. All right, down here. So I'm making a little table of the proportions rather than the raw counts, right? And we did this way back at the beginning of the semester when we had absolute frequencies. Those were the counts when we were doing probability simulation. And then we divided that by the total trials or randomizations to get the proportion or probability of out of all of the trials, right? So that's exactly what we're doing here. We want to find the probability rather than the actual count. All right, so 4715 divided by 5087, that's going to give us 0.93. And the helmet row should add up to 1. Same thing with the no helmet row. We're going to do 267 divided by 1658. That's going to give us 0.16, and we're going to do 1391 divided by 1658, and that gives us this 0.84. All right, make sure you understand that. And in the next slide, I'm going to show you how the graphs look, the count graph versus the percent graph. All right, so like I said before, we want to compare the absolute frequency, the count, the actual raw data, how many people did we find, versus the relative frequencies. All right, relative frequencies, we're going to divide by the number that we got in each of these categories. All right, so here we saw, strangely, that it looked like there were fewer head injuries than the no helmet group. But when we look at the percents instead of the counts, we see, aha, lower head injuries compared to helmet versus non-helmet, and other injuries are higher in proportion because they have to be. So we see kind of a reverse pattern of what we saw. Now, there were more other injuries in this other graph, but it's really not as much more different as it looked like on the original graph. Okay, so when we have count data and we have different numbers of individuals in our categories that we want to compare, showing the count data can be very misleading, all right? And you want to show these data in a percent, relative frequency, and that could be interpreted as a probability. All right, so here we see overall our conclusion from this graph is that overall other injuries are much more common than head injuries are, but that head injuries, you have a lower probability of having a head injury if you have a helmet on compared to not. Now, these percents are still fairly small, right? So um, those are actually probabilities, not percents. So if we do call them percents, we have what, a 15% of, if we have no helmet, we have maybe 8% if we have a helmet on. And so you could say that you're twice as likely to have a head injury with no helmet as with a helmet, but it's not a huge percent. You aren't going from 20% to 40% or 50% to 100%. Right. So different ways we can describe what we're seeing on this graph can give it a different, make it sound more dramatic or less dramatic than what we see on the graph. All right, so there's another thing I want to show you about how the graph looks when we decide to put the one of the variables on the legend versus along the x-axis. All right, so in this graph, this is the same one, the count graph that you've seen twice before. All right, so we've got helmet versus no helmet in the legend. I remember that we specifically wanted to do this so that we could compare helmet to no helmet. All right. Now, on this version of the graph, I switched the row and column variables. All right. So now I have helmet being put on the x-axis, helmet versus no helmet, and head injury versus other injuries on the legend. So now we have cyclist behavior or cyclist safety choice on our x-axis. And what's easy to do in this version is to compare head injury to other injury, right? Because they're right next to one another on the graph. And you think to yourself, oh, wow, whether you have a helmet or you don't have a helmet, you have a lot more other injuries than you do head injuries. But you might conclude from this graph, just like we did with the counts, that you have a lot more injuries overall in if a rider wears a helmet. Like somehow it makes them think that they're invulnerable and they ride like crazy people. All right, but no, that's simply because there's a lot more people using helmets, and so there's just bigger numbers for both of the helmet categories. Okay. So, so that's something to keep in mind when you're making these graphs. Just make sure that you have that variable you want to compare in the rows. All right. When you're making the graph in Excel, it will automatically put that for you into the legend. All right. So our question that we're asking, we're going to look at those data and we want to compare something about helmet versus no helmet, and we specifically want to look at head injuries, right? So we're asking the question, is a cyclist wearing their helmet or not, independent of the type of injury they get, right? So we want to ask the question or we want to look at how we think about this question using the rules of probability. If the two variables are independent, then this mathematical rule will apply. Remember, we used this equation earlier in the semester when we were using dice and cards and those kinds of things, which we know are independent from one another. Probability of A and B is going to equal the probability of A times the probability of B. All right. Now, the reason we have to specify that if the variables are independent here is that if the two variables are not independent, then the probability of A times the probability of B is not going to equal the probability of A and B. right? We actually need to measure it, right? So we need to go out, we need to find the individuals, for example, who wore a helmet and did not get a head injury, right? So that would be probability of A and B. We actually have to get the data and look at it, all right? So often it seems a little circular, right? We're going to have a reason that we think these variables might be related to one another, all right? So we don't actually think they're independent, or we don't know they're independent. Okay, so we're going to start by assuming, um, we have to check it. We can't just in real world examples, not like dice and cards and things that really function like those model probability devices. Most of the time, we are going to have to start by figuring out whether those two variables are indeed independent or not. All right, so how are we going to know that? How do we know? Well, we're going to check this questions, right? So we're going to actually ask, if I measure the probability of A and B, right? And I also separately measure the probability of A and the probability of B, and I multiply those two things together, right? Just like the mathematical rule tells me we should be able to. Do they equal one another, right? So we're going to use this strategy in our test for independence using the chi-squared test statistic, right? So that's a logic of we're going to measure these things and we're going to ask the question, do they actually equal one another? Okay. If they do equal one another, then our two variables are independent. If they do not equal one another, then our variables are, in fact, not independent, right? And the structure of how we actually do that comparison, what we calculate and calculate the chi-square test statistic, and is there some difference between the probability of A and B? And if we multiply the probabilities of A and B separately, like, is there a little difference? Is that difference big enough that we want to reject that null hypothesis? Okay. All right. So there's a little bit more probability math that I want to show you and talk about because it will make the calculations that we eventually do make sense. It's going to seem kind of confusing when we are first going through it, though, but bear with me. All right, and I want to use an example of crayfish. So crayfish are small crustaceans. They're kind of like little lobsters and they live in freshwater. And so they're more like hand-sized rather than uh, small dog sized like lobsters are. All right. And so suppose we did an experiment where we set up containers, little aquariums in the laboratory, and we had a little hiding place for the crayfish in each one, a little crayfish house. And then we hid food around in the tank, and we randomly picked, we put them on a daylight, a dark light schedule, and we randomly picked each of the crayfish to sample or check what it was doing during a dark or a light. So we had, 123 crayfish total in this experiment. And we just checked, okay, is it darkness or is it light in our experiment? And is my crayfish hiding or is it out foraging, looking for food? Okay. All right, so kind of time-consuming experiment. Lots of little crayfish and tiny houses that they can hide in. All right. So this these tables that we're used to seeing already, that we were just talking ad nauseum about making graphs from, if we suspect that these two variables have a relationship with one another, we're going to call it a contingency table. All right, contingency, I'm going to define that for you in the next slide. All right, so we've counted up individuals that we saw in each of these combinations of characteristics. So in this experiment, we think that the dark and the light conditions may have some effect on the crayfish behavior. Right, so dark versus light is going to be our light condition. That would be the overall variable here. And hiding versus foraging, we could think of it as the behavior of the crayfish. All right, so if our question here is behavior independent of light condition, all right, that's the question we're asking. All right, so what we want to do is look at, does the probability of, for example, right, we've got 4 different cells in the table here, and we want to know if it's true for each of those 4 cells in the table. Right, but I'm showing you one as an example. All right, does the probability of dark and hiding from the table, the number of individuals here compared to the total, does it equal the probability of dark from the table times the probability of hiding? All right, so we can find each of these probabilities from our table. The probability of dark is going to be how many total individuals did I have that I recorded their behavior in the dark. All right, so when we do our row totals for our contingency table, then that's how many individuals were sampled during the dark hours. And then how many of them did we find hiding? That total in the experiment is this value 66. All right, so when we want to find the probability of crayfish, when we peeked at it, was it in the dark? That's 50. And how many is it out of? It's out of how many total crayfish there were in the experiment. Okay, so the probability of dark, or being in a dark condition, is going to be 50 out of 123. Now this value 123, that's our grand total of how many crayfish there are in the experiment. And we find that just by adding up all 4 cells of the data table, or however many cells there are, or if you want to take a little shortcut, I like to add up the total of the two of all the rows. Okay, you can also add up the total of the columns, but what you don't want to do is add up the total of the rows to the total of the columns. That'll give you twice the sample size. All right, so you just total up the rows, and that gives you the total, or the grand total. All right, so we've got 50 divided by 123, that's 0.41. We've got 66 divided by 123, that's 0.54. Now, when we look at how many individuals are actually in this dark and hiding square, it's 18. All right, so 18 out of 123, that's 0.15. And so that's our actual measured probability of dark and hiding. All right, so our question is, if they were independent, then the probability of dark times the probability of hiding would equal the probability of dark and hiding together. So if we multiply the probabilities of dark and hiding, we do 0.41 times 0.54, we get 0.22. All right, so I've highlighted these values of the one we calculated from the probability of dark times the probability of hiding. All right, so if they were independent, then that is what we should find. 0.22 is the probability. But what we did find of measured individuals was 0.15. All right, now 0.15 is pretty different from 0.22. All right, but this was only for one cell in the table. Okay, so we want to have a method where we can ask, how big is this difference? Is it big enough that we want to say it's significantly different? And also we want a way that we can incorporate the data from all of the cells of the table, not just one at a time. All right, so mathematically, what we are finding is called the conditional probability, right? And the contingency table is specifically designed to be a way to help us find the conditional probability. So if we know that the state of A, if whatever this variable A is, changes the probability of B, then those two variables are not independent. If we know the probability of A and B, like we did in our table, then we can actually calculate the probability of B conditioned on A. All right. So we're also going to say that B given A, and the notation for writing it is capital P, and then in parenthesis, our capital B, and then this vertical line, I think it's called type in computer programming, and then the A goes after it. And then so that B vertical line A is what we're finding the probability of. So what this means is important to understand. So, and I'm going to use an example to help you understand. Okay, suppose I asked you, what's the probability it will rain tomorrow in San Diego? All right, now I'm recording this the second week of October, and it's actually already, we had a pretty serious rainstorm a few days ago. But we are in the season of the year where it is indeed known to rain, also called winter. All right. So if this were a day in the summertime, then I would say, well, there's essentially zero probability of it raining right here in San Diego. It just doesn't rain in the summer. There's a couple weeks in August that it might rain a little bit, but typically, it doesn't. Right. So we have drastically different probabilities of having rain out of different times of the year. All right. So if I ask you a question, what's the probability of getting rain on a given day in San Diego? But I don't want to know at all what season it, like you don't know what season it is. All right. So I looked up the data from the National Weather Service, and it actually rains in San Diego um, on average 41 days per year. All right. So 41 days out of 365 days is 11%, right? So if you didn't know what day out of the year it was, and I asked you, what's the probability of it raining, right? Then that's the only answer you could give, right? You don't on any day, right? So that's 11%, right? So that's our probability of B. All right. So rain is the probability of B. All right. If we know the state of A, so by that I mean if we know what season of the year we're in, okay? So now I tell you it's winter, all right? And by definition, winter is from December to March, all right? And in winter, we get rain on average 6 days per month. All right. So on any day in winter, we actually have a probability of rain of 20%. All right. So 6 days out of 30, or approximately December, January, February, March, 4 months. So we multiply 6 times 4. That gives us 24 days. We've got 4 months. That's about 120 days. So 24 divided by 120 is 0.2 or 20%. Okay. So that's an example of this condition, a conditional probability. What's the probability of rain given that we know that it's winter, all right? So that's going to have a different probability than the probability of rain if we aren't specifying any condition, all right? We didn't specify we're in San Diego. We didn't specify what season it is. All right, so that's the idea of this conditioned. All right, and what we do is in this notation, P, the probability of B given A, all right, the A thing has to happen first. All right, so it's a little backwards in time of what? All right, so the probability of B given A. Now, we can find the probability of B given A using this formula. The probability of A and B that we've measured divided by the probability of A that we also know. All right. If they're independent, you'll actually get this value, probability A and B, is going to be equal to probability of A times probability B. Probability of A will cancel out, and you'll just get probability of B. All right, so if you don't know if things are contingent, but you have these values, you'll just get probability of B as the answer. All right. So this is a formula we can use. This is the way that these conditional or not independent kind of relationships will be written in probability mathematics notation. All right, so we can think of the term conditioned on, we can replace it with contingent on. All right, so that the way we talked about our data table being a contingency table is basically means the same thing. It's a conditioned on, or is it contingent on? All right, is something going to change if this first condition is satisfied. All right. All right, so let's do a quick calculation of a um, of a an example of what we were just describing, this probability of something given something else. And we'll use our crayfish data again. All right, so if we know already, or we assume that our behavior variable and our light conditions are not independent, and we want to know the probability of H, or hiding, given D, all right, then we can find it using this equation, the probability of D and H divided by the probability of D. Right. So we need to find this probability D and H. All right, that's the same as we did before, probability of being in this dark and hiding at the same time. All right, so 18 out of our total 123, same value, 0.15 that we got before. Now we need the probability of dark. And that's the same as we calculated before, looking just at the row for dark. How many crayfish did we find in dark conditions total in the experiment? 50 divided by 123. That equals 0.41. All right, so if we're going to take, and I'm showing the fractions for a reason here in the calculation. Now, often you'll just want to do these calculations based on the decimal numbers. It's easy to do in your calculator, but look carefully at these fractions. All right, 18 over 123. It's our probability dark and hiding. We're going to divide by the probability dark. All right, and that's 50 over 123. So here's this fraction. 18 divided by 123 over 50 divided by 123. All right, so when we divide by a fraction, this tricky algebra or fraction math, which nobody ever likes, all right, when we divide by a fraction, it's the same as multiplying by its inverse. All right, so what that means is I'm going to take 50 over 123, and I'm going to flip that. So I'm going to have 123 over 50. And instead of dividing, I'm going to multiply. So this converts to 18 over 123 times 123 over 50. All right. And so I've got 123 in the denominator of my first term, and I've got 123 in the numerator of my second term. And that means they'll cancel out. All right, so 18 divided by 123 times 123 divided by 50 is going to equal 18 over 50. All right, 18 over 50, look, we can read it right from our table. All right, so if I want to know the probability of hiding given dark, we're saying, okay, I know these crayfish are in a dark condition. What's the probability they're hiding versus foraging? Right, so what this contingency table allows me to do is basically see, given dark, what's my probability hiding? Given dark, what's my probability foraging? All right, so we're going to isolate this row, and we can just calculate 18 out of 50. All right, so if we have our data set up in this way, then our contingent probabilities are really easy to find. Okay, so, and it also helps you understand what does that contingency mean. It means, well, we're only going to look at the ones in this dark category. All right, we could do the same thing. We could do the probability of foraging given dark, right? And we just do 32 out of 50. We could do the probability hiding given light, all right? Say we have a crawfish that's in a light condition. What's the probability it's hiding? All right, that is also a contingent probability. And we'll just look at this row and we'll say, oh, it's 48 out of 73. Okay, so that's really a shorthand for going through all of the steps that we would do in order to make this long calculation. Right. Okay. So I hope you understand that. And we're going to use these different calculations. So we've been practicing the calculations that we're going to make when we're doing an actual hypothesis test for the chi-squared test of independence. All right, so remember when we're doing a chi-squared test, we need to have observed values and we have expected values. In the chi-square test for goodness of fit, we had expected values based on something that we planned ahead of time, whether it was proportions based on Mendelian inheritance, or whether we thought things were going to be equally likely, or we had some reason for saying, you know, I want to see if my data matches this pattern. All right, so our expected values were something that we just calculated based on a prediction. Our expected values for chi-square tests for independence are going to come from the data themselves. All right, so in our test for independence, we're going to have a null hypothesis that these two variables are independent from one another. Right, so behavior is independent of light conditions. Right. So our knowledge of probability math tells us if these variables are independent, then for all cells in the table, the probability of A times the probability of B should equal the probability of A and B. All right, so we've done these calculations, we've done the multiplication for a couple of examples already. So that should be true if our values, if our variables are independent. All right, so we're going to calculate our expected values, right? Because remember when we calculate our chi-squared statistic, we're going to take the sum across all the cells of the observed minus expected. We're going to square that difference and then divide by the expecteds. Okay, so we need those observed and expecteds to compare. All right, so under the null hypothesis, if behavior and light conditions are independent, then our observed data, right, our observed data is going to be the number of individuals we did find in each of our four conditions. All right. If they are independent, then those numbers should equal the ones that we'd expect based on multiplying their probabilities together. All right. So if they're independent, our expected values can be calculated by doing probability A times probability B for each of the cells in our table. Now, those are probabilities, right? Those are going to be numbers that are less than 1, decimal numbers. So in order to find our expected values, right, we have these counts. We don't have probabilities in this table. We have 18 crayfish, right? So what we're going to do is we're going to take the probabilities of getting each of these combinations, and we're just going to multiply it times the number of total individuals in our experiment or our survey. Okay, and that's kind of like we did in the expected values, right? We said, oh, I'm expecting a proportion of 1 to 2 to 1, but my probability for, so my probabilities are 0.25, 0.5, and 0.25, but I've got 20 individuals that I measured in my experiment. So I have to multiply 0.25 times 20 to get my expected value. All right, so the expected value is going to be the number of individuals you expect to find in that condition, assuming the null hypothesis is true, based on the number of individuals you have in your overall experiment. Okay, so let me show you how this works in this example. All right, so what we're going to do is we're going to start by using this to find the probability that we'd expect if the null hypothesis were true, probability A times probability B, right? Then we're just going to multiply it times the sample size. Okay, so I'll start with the same one that we've already done. Probability of dark and hiding, right? Probability of dark is going to be 50 out of 123, just like we've calculated it before. And we're going to do the probability of hiding is going to be 66 out of 123. Now, I would just do these as fractions or as decimals, but I want to show you something. Okay, now we're going to take that probability and we're going to multiply it times 123, the total sample size, all right, to turn those probabilities into accounts, all right, numbers of individuals. All right, so I've calculated these values already on previous slides. We've got 0.41 times 0.54 times 123. All right, now that gives me 26.8. Now I can't have 0.8 of a crayfish, but because of the way you calculate the expected, you're going to get decimal numbers for these, and you should use them. All right, you want to use the decimals. We're not going to round those off. Our expected values are not actual crayfish, they're hypothetical crayfish. All right, literally, if our null hypothesis is true, this is how many crayfish we should have. Okay, so I want to show you something, and that's why I wrote it as fractions, right? We're multiplying 50 times 123 divided by 123 times 66 times 123 divided by 123, sorry. And then we're going to multiply that times 123. All right, now that means we can cancel out, all right, this 123 that's going to be in the numerator with one of these 123s that's in the denominator. Make sure you see that. All right, and what we're going to end up with is 50 times 66 divided by 123. All right, so there's a simplification of this. You don't have to actually do the probabilities and then multiply them together and multiply times 123. There's a calculation shortcut, all right? And when we write it down, it's hard to write it as an actual formula. But if you're a visual learner, it's not so hard. And you can calculate the probabilities like this every single time if you want to. You can find the probability of dark, find the probability of hiding, multiply that times the count. All right, you are perfectly welcome to do that. a shortcut, a lazy/smart shortcut. We're going to take the total for the row of that cell. We're going to multiply it times the total of the column of that cell. We're going to divide by the total. All right, so we kind of have a little visual pattern. To find the expected value in any cell, you're going to take the row total times the column total and divide by the total. All right, so we did that already for dark and hiding. So next we're going to go into the foraging column and we're going to have the same, it's also dark, so we're going to have 50, but instead of 66, we're going to have 57. All right, the sum of the foraging column. So we're going to have 50 times 57 divided by 123. That gives us 23.2. Now moving to hiding and light, we're going to have 66. Oops, I'm going to do the row first. 73. All right, we don't have a 50 anymore because we moved to the other row. 73 times 66, because we've got light and hiding. All right, 73 times 66 divided by 123. All right, they're all always divided by the total. All right, and then our last cell, 25, is our foraging and our light count. All right, so we're going to do the row total, 73, times the column total, 57, and then we're going to divide that by 123, the total. All right. So make sure you're understanding that I have these titles of my different categories, hiding and foraging. I've highlighted them in gray for you to make sure that you're seeing the difference between the categories. All right, and then we have this column for the totals. So we can also refer these to these as the marginal totals, the totals that are at the edge of our contingency table. All right, so this table is actually a 2 by 2 table. All right, it's got 2 columns and 2 rows. All right, so it's a 2 by 2 table. So those are all of the cells in the table that we need to calculate an expected value for. All right, so far this lecture may seem like I've been going over the same thing, weirdly calling it different things. All right, but hopefully when we get to our hypothesis test steps, you will understand why this has all been worthwhile. All right, so if we want to do our chi-squared test for independence, hypothesis test, we're going to start with the hypotheses. Our null hypothesis is always going to be, and it's going to be written in words. We don't have symbols to use for this. So row variable is independent of column variable. Now you, the thinking scientist, need to substitute in the name of that row variable and the name of the column variable, right? And then the alternate hypothesis is the row variable is not independent of the column variable. All right, so if we can reject our null hypothesis, they are independent. Then we can accept our alternate. Ooh, they're not independent. All right, these two variables have some relationship between them. All right, we're going to need to have observes and expecteds in order to calculate our chi-square test statistic. Our observed counts are our data. All right, we observed it. Our expected counts are calculated what we should have if our null hypothesis is true, right? So those row total times column total divided by the grand total calculations that we just made, those are going to be our expected counts, right? So they're derived from finding the probabilities, assuming they're independent, but we have to convert them into counts so that we can actually compare them to our observed counts, right? And you may get decimal place values for your expecteds, and you don't want to round those off. You want to use the decimal values. I mean, round them off to a couple of places. All right, so if you remember from the chi-square goodness of fit test, we need a degrees of freedom to use this test, right? And for chi-square test for independence, the degrees of freedom is called nu. This Greek letter kind of looks like a V going fast. And it's going to be the number of rows minus 1 times the number of columns minus 1. So if we have a 2 by 2 table, we have 2 rows minus 1 is 1. We have 2 columns minus 1 is 1. We're going to get 1 degree of freedom. Okay, so in a 2 by 2 table, we only get 1 degree of freedom. As we get bigger tables, we get more degrees of freedom. The reason we need to do that is because as we're adding up the observed minus expected squared over expecteds, that number, the sum, the chi-squares, is gonna be bigger just because we have a bigger table. So the shape of the chi-square distribution changes to take that into account. All right. So we're going to calculate the chi-squared test statistic. We're going to find the p-value using our chi-square distribution table. We are going to, for whatever alpha level we have, we are going to reject or accept the null. If our p is less than our alpha, we're going to reject the null. And then we're going to write our conclusion. Remember, if you reject the null, you're going to go back and say, aha, I reject the null. Therefore, my row variable is not independent of my column variable. All right, and typically for these chi-squared tests for independence, we'll have made our graph that's showing the pattern in our data, and then we want to look at it and say, oh, what is the pattern? What is this non-independence? Okay, so I am now going to work an entire sample problem for you of the whole hypothesis test, and I have some nice data on is algal symbiont color independent of water depth in giant clams. All right, so if you imagine a clam, a bivalve mollusk like a clam, a mussel, a scallop, the shells of these giant clams have this wavy edge, and they are giant compared to clams, but they're, and the largest species, Tridacnogaigus, can be over a meter wide, right? They're not as giant as you sometimes see them in the cartoons. And when the animal is alive, it has its mantle, or the kind of, maybe you see a snail crawling and it has this big um, like the mushy part that's inside the shell. What we see in a giant clam is it actually has tissue extending across the opening of its mouth, right? So it doesn't really look much like a clam, and they sit with the mouth of their shell open, just kind of on the seafloor. And this tissue of the clam is embedded with, just like coral animals, it's embedded with a symbiotic single-celled algae that has photosynthetic pigments, right? So giant clams are these huge animals that are living in coral reefs, which are very poor in plankton in the water. And so they have also evolved just like coral animals, a symbiotic relationship, a mutualism with these single-celled algae. And so they're embedded in the tissues, and that makes these giant clams look all glowy and bright, beautiful colors when you see them and they're actually alive. And these algal symbionts, there's different types of them, and they actually can have different photosynthetic pigments that look different colors, just like you see macroalgae like kelp that's brown versus red, different kelps and algae or green, right? So we have these different colors, and this image is showing green, and then this sort of brown and aqua, really beautiful colors. And off to the side, you can see a blue one over here. All right, so we're curious if the depth that the giant clam is living at is going to influence the color of the algal symbionts, because as sunlight goes down through the water, different wavelengths of the sunlight are dampened, or they disappear. Right, so if you go deep enough in the water, everything's dark. But when you're in very surface water, all different colors are bright. But then as you go deeper, some of the colors are removed from the color spectrum. So different algal symbionts have different photosynthetic pigments, different colors that actually are better at absorbing different colors of light. So we might think, well, we should see colors of the clams that match the depth of the water so that the photosynthetic pigments are most active. So that's our question. It was a little complicated to explain, but it's a neat example. So I wanted to show you these pictures and give you that explanation because it is a life sciences statistics class. All right, so let's look quickly at the data and the graph for this example problem, and then I'll transition to the paper and pen and show you all the steps. So here is our data table. We're curious about what the effect of depth is. So it's acting like our independent variable. And we've just separated into shallow, less than 5 meters, versus deep, greater than 5 meters. And some poor scientists had to go around snorkeling in these beautiful tropical waters and counting up the giant clams that they found and what color symbionts they had. And so we've got our data here. Now this table is actually 2 rows and 3 columns. All right, so make sure you're noticing how that works. 2 rows, 3 columns. And that we have a total of 83. And when we graph the data, all right, notice on our legend, we've got the rows variable. We've got less than 5 meters, greater than 5 meters. We've got the 3 different algal symbiont colors. We've got brown, we've got green, and then there's individuals that have both brown and green. Huh? Maybe the best of both worlds. All right, so notice we have a little bit difference of proportions are just, I did these as raw numbers because there's not that much difference between our numbers we found in the depths. So it looks like we have a little more brown and green in the shallow and a little more brown and green too. Sorry, brown separately, green separately. We have a little more brown and green together at the deeper depths. Are these differences between these 2 rows of data in our table? Are these differences enough that we're going to say there's a significant difference? So let's work this out and see what the answer to that question is. All right. So our overall research question is algal symbiont color independent of water depth in giant clams. So we have our two-way table or our contingency table. We've got our independent variable, the one we think is causing the other one to happen, depth in our rows. All right, and we have 2 rows. So I'm going to write down little r equals 2. We'll need that for our degrees of freedom. And we've got our algal color across the columns. We've got these different colors, the 2 colors, and then the combination of both. All right, so we've got 1, 2, 3 columns. I'm going to write down C equals 3 because we'll need that for our degrees of freedom as well. All right, so these are our observed values. We get those are our data, and then we're going to use them to calculate our expectance. All right, so I'm going to highlight these and then highlight where we put them into our chi-squared equation, O for observed. So we need our expectance. So the first thing that we're going to do is we're going to write down our null hypothesis. All right, so when I gave you the general way that we write the hypothesis, I said row variable is independent of column variable. So here's where you want to substitute in, instead of row variable, the actual row variable. Okay. So water depth is independent of algal symbiont. And so our research question is a question. Our hypothesis is a statement. We're saying it is, right? Versus our alternate water depth is not independent. Oh, that should be up. Up. Yeah, water depth is not independent of algal symbiont color. I am going to set our alpha to 0.05. And what else do we need? Oh, we're going to need our degrees of freedom. And that's going to be little r minus 1. And little r is the number of rows that we have. And we're going to multiply times little c minus 1, number of columns. Now you can also think of it as you're going to kind of block out one row and one column and see how many you have left over. So according to the formula, we're going to do 2 minus 1 times 3 minus 1, which is going to equal 1 times 2, which is 2, which the finger method also got us. All right, so those that we are going to need. And then most of the work is calculating the expecteds, right? Now, calculating the chi-squared is a little bit of work too. So I like to make myself a nice big table that matches the shape and size of my data table in order to my expecteds, because we've got a lot of numbers going on, and when we do the subtraction, we want to also be really clear that we're doing the subtractions with the correct expected. So it's just a easy way to make sure that or to help you make your calculations the most accurate. All right. So our little equation, our formula for the expecteds is that we're going to do, so we want to start with brown and less than 5 meters. All right. So I'm going to do the row total 44 times the column total 47 divided by the total 83. All right. So I'm going to calculate that with my calculator. And you get 24.9. All right, I'm going to work across. So now I am going to do this cell in the table. So I've got the row total times the column total, 12, divided by the total, 83. And I get times 6.4. As you're starting to make the calculations, you can see whether they are kind of matching if your expecteds are kind of matching your observed. So far, they're close. All right, so then I'm going to do the third one across, and I'm going to do 44 times 24 over 83, and that equals 12.7. All right, now I'm in the second row, so I'm going to go to the second row, and I'm going to have 39 is now my row total times 47 divided by 83, and that equals 22.1. And I've got two more. I'm going to do 39 times 12. 6. All right, so as you're going across, you're always going to be using the same row total, and as you're going down, you're going to be using column total. So once you get your expecteds table started, expecteds, these are our observed. Once you get it started, you can actually, you don't have to keep looking up here because I will know that this one is going to be 39, like my other ones in this row. And then the column is going to be the same as. And that calculates out to 39 times 24, 11.3. Okay, so these numbers are my expected values. We're not done. All right, so in order to, now we have our expecteds, we've got our observes, we've got our expecteds. The next step is to calculate our chi-squared test statistic. Chi-squared equals, and then I'm going to sum up the observed minus expected squareds divided by the expected. I'm going to have 6 terms in my equation. And like I said before, it's going to be easy to write down my observes minus my expected since I have them in a matching format. All right, so 26 minus 24.9. We're going to keep these decimal places because the expecteds can have them. Divided by expecteds. So the second term, you're going to just copy it down. There's my first term. Now I'm going to have 7 minus 6.4 squared over 6.4. 11 minus 12.7 squared over 7. Again, I'm going to actually write my chi-squared formula on the paper in the same pattern as my observes and expecteds are. 21, that can help me make sure I have all the terms. 21 minus 22.1 squared over 1 plus 5 minus 5.6 squared over 6 plus 15 minus 11 squared over 11.3. All right, so you can see that these calculations are going to get kind of tedious if we have a very large table. So we're typically, like always, going to have software do these calculations for us, but I do want you to practice doing at least a couple by hand. Again, we're building our skill at substituting into the formula so we understand what the formula is doing. Are building our skill at being meticulous and careful, which even when you're using software to do your statistical tests for you, when you're entering the data, when you're selecting different choices in the software package, you need to have the ability to focus, concentrate, and make sure that each and every detail of your work is correct. All right, let's see. Okay, I'm going to add all those up. So make sure that you calculate out each term and that you try and add up and make sure that you get approximately the same number as I did here. I've got 0.73. All right, 0.73. That is the value of my chi-squared. All right, so what do we want to do with that? We're going to take our degrees of freedom and our value of chi-squared. Now we're going to go to our chi-squared distribution table. All right, and so our degrees of freedom is 2. 0 is 2, so we're going to go across this row. We are looking for our chi-squared value. Oops, our chi-square value 0.73 is less than 2.77. All right, so I'm going to write down our chi-squared is less than 2.77 for p-value is greater than 0.25. Right, so we go to the top here and see, and then again, as our chi-square values are getting smaller to the left on the table, p-values are getting larger as we go to the left. All right, so I wrote that down. Chi-squared is less than 2.7 leads us to a p-value that is greater than 2. Is p less than our alpha? It's the same question. The answer is 0.25 compared to 0.05. P less than our alpha. Oh, it is not. Okay. So in this case, we do not reject null. Now, we're not done. All right, we didn't reject the null. So what's our conclusion? Well, we accept the null. Water depth is independent of algal symbiont. All right. So based on the data we collected, the depths that we used, the colors that we looked at, we didn't find that there was a relationship. Now, we could have designed our experiment differently. We might get a significant result if we included more colors, or if we um, included finer gradations. All right, but remember looking at the graph earlier, go back and look at the graph, and there weren't a huge differences between the number of clams in that had each of the color combinations in the depth. In the two depths, right? So in this case, those differences, they weren't exact, right? Our observed and our expecteds weren't exactly the same, but there wasn't enough total difference across all of those groups that we could reject the null. So, you know, our color and our depth are indeed independent based on this experiment. Okay. All right. So you can just sort of memorize the steps of doing the equations, but I think the explanation of where these probabilities come from and why we're doing what we're doing in order to really understand what we're doing with this test, I think that's worthwhile. Um, but you should at least be able to work through problems, even if you don't really understand all of the probability steps that we did. All right, great job. I'll see you next time.